Hello, general biology students. I hope you have enjoyed the snow day on Thursday. As I promised on Tuesday that we would be sure to talk about the topics that you told me you were most confused about on Tuesday, and then Thursday we had no class. So to be able to accomplish that goal, I am recording these two videos. This one is going to talk about catabolism, and so we'll specifically be talking about Krebs cycle and electron transport chain and chemiosmosis as it relates to the electron transport chain. I'll be making a second video which will cover photosynthesis and your questions there. So look for both and let's get started. So just as kind of a review and connecting us back to the previous topics, we talked about the laws of thermodynamics and how energy could not be created or destroyed, it just changed form. And so you can see in this diagram that we do not have a closed system in an ecosystem. We have light energy coming in from the sun, and we have heat energy being lost from that ecosystem. Now, some of the light energy, as you can see, is being captured and changed into another form, and that form in this case is ATP. So, let's talk about the steps of catabolism of glucose in an aerobic environment. Okay, so we've already talked about that there are three steps. We have glycolysis, we have Krebs cycle or citric acid cycle. They are one and the same, just two names of the exact same thing. And then we have this electron transport chain, which is our third step that we talk about in an aerobic environment. Now, if you remember, we said that the overall reaction was glucose plus oxygen gives us carbon dioxide, water, and ATP. But if you remember, these different reactants and products are coming from different steps in these three processes. Glycolysis is taking glucose, and we're ending up with an end product of pyruvate. And after that's oxidized, it feeds directly into the Krebs cycle or the citric acid cycle. But remember, since this is a cycle, it goes in a circle, and we take one of the last steps and input our acetyl-CoA, which is what we get after we oxidize pyruvate, and we turn that cycle. And it is throwing off electron acceptors, both NADH and FADH2, and off also our carbon dioxide molecules. We then pass these electron carriers to the electron transport chain, which, being coupled to chemiosmosis, we make lots and lots of ATP. So let's look at some diagrams. So here's just an overview of what we just looked at. Notice that we are making ATP in all three of these steps. However, we make significantly more ATP over here. We only make a couple of ATP by substrate level phosphorylation in glycolysis and the citric acid cycle. Okay, so in glycolysis and the citric acid cycle, we use substrate level phosphorylation. Again, enzymes are showing up. That's why we talked about them so much before. And in this case, we are able to steal a phosphate group from a substrate with the help of an enzyme and put it on an ADP and make an ATP. As you can see here, this is happening in 7 of the 10 steps of glycolysis, but it's also happening in some of the steps of the Krebs cycle. So, if no oxygen is present, we do fermentation. The importance of this is that fermentation does use pyruvate to give you a lactic acid. However, the main thing that needs to happen is that we need to recycle and get back to some NAD pluses. We need to recycle our electron carriers. If you can't recycle the electron carriers, then glycolysis stops. And in that case, you can't generate ATP at all. So while it is important to break down the pyruvate and try to harvest the energy from it, the most important thing when we're talking about fermentation for our purposes is to understand that we're also able to recycle these electron carriers so that they can go back to glycolysis and again be reduced. Okay. That's just what I said. Fermentation is recycling our electron carriers. Remember that fermentation is happening in the cytosol. It is not happening in the mitochondria. Again, we are releasing CO2 just like we would during the Krebs cycle. But in both cases, we are reducing NAD to NADH. Once we get past glycolysis and fermentation, acetyl-CoA, if we're going to continue in Krebs cycle, electron transport chain, and use chemiosmosis to make ATP, that is all going to happen in the mitochondria. And remember how we talked about the inner membrane being folded, and that was important because it increased the surface area of that membrane, which allowed more of our enzymes, whether it's ATP synthase or our other ubiquinone, coenzyme Q, those coenzymes and other enzymes that take place in electron transport and chemiosmosis, we can fit more of them in the larger surface area of the folded inner membrane. Okay, so let's get right into the citric acid cycle. As we have said, we take pyruvate from glycolysis, and notice we get 2 molecules of pyruvate from 1 glucose, which means that each molecule of glucose will turn the citric acid cycle 2 times. So we take the pyruvate, we add a coenzyme, and there at that point we generate our first electron carrier. Acetyl-CoA can then feed into the citric acid cycle where we take off the coenzyme and we begin to turn the cycle. Now this picture isn't showing you all the steps. Our next one will show you all of the steps. But notice we are doing substrate level phosphorylation here. And we are creating 1, 2, and 3, so that's a total of 5 electron carriers in one turn. Notice that we need water, so water goes in, acetate goes in, and our oxidized electron carriers. And actually, yes, that is what we have. I'm sorry. Okay, so here is the scary sheet that shows you all of the steps of the citric acid cycle. I am not, as I said before, not going to ask you to memorize all of the steps. As you go forward in other biology classes, and even in this class, we're going to use our knowledge, though, of the citric acid cycle in different ways. For instance, later on, we are going to use a substance that will substitute or act like FAD in lab, and it will actually steal electrons in a step. And so, therefore, you can measure the rate that succinate is being converted to fumarate in this case. In microbiology, we actually have a media that has oxaloacetate in it and citrate in it, and it'll actually have a color change as citrate gets converted to either isocitrate and ketoglutarate. And it's very easy to see if the citric acid cycle is actually happening. So again, you're not going to be required to memorize all the steps. However, it is important to know what goes into the citric acid cycle and what we get out of it. But because it's a cycle, we're not getting any molecule per se out. We are only getting ATP, carbon dioxide, and electron carriers out of this cycle. So acetyl-CoA goes in, and we end up, as we said earlier, with an ATP with electron carriers, both NADH and FADH2, and also ATP. We also said, and CO2. I already wrote down ATP. This is the main thing to know, that our electron carriers are then going to go to the electron transport chain. CO2 is excreted. This is where the CO2 that you and I breathe out comes from. And we also make a small amount of ATP. Okay, as we have said, really the most important thing that we get out of the Krebs cycle are the electron carriers, NADHs and FADH2s. So those, remember, are holding energy in those electrons. But it's not a form that the cell can directly use to power work. So to actually efficiently use glucose, we need to couple the oxidation of NADH and FADH2 to another enzyme. So what they're going to do is they're going to donate their electrons to an electron transport chain, and that electron transport chain is then going to power chemiosmosis, which is where our ATP synthesis comes from, using oxidative phosphorylation. So we're going to see a picture in just a minute where we can simulate this happening. As our donated electrons from the NADH and FADH2 go through the electron transport chain, that is going to fuel active transport. So as those electrons fall back down to their ground state, the energy that is released from that process powers active transport, where we pump protons, or hydrogen ions, if you will, across the inner mitochondrial membrane back out into the intermembrane space, still within the mitochondria, between the outer membrane and the intermembrane. That creates a concentration gradient of hydrogen ions, where we have a higher concentration in the intermembrane space than we do in the inner membrane space. And then ATP synthase is going to act like a turbine as those hydrogen ions travel back down their concentration gradient, and that will fuel the formation of ATP. And so we call this whole process oxidative phosphorylation. Again, anytime you see phosphorylation, we are almost always talking about the creation of ATP. Remember, in glycolysis and Krebs cycle, we had substrate level phosphorylation, where we stole a phosphate group from another substrate using an enzyme to make ATP. Here we are using ADP and inorganic phosphate to make ATP, driven by the fall of the hydrogen ions back down their concentration gradient. So let's look. Okay, this picture is representing electrons that are being passed from our electron carriers down the electron transport chain. And as you can see, there are multiple subunits that are involved here. So basically what is happening is each electron gets passed from this flavin molecule, and we also see iron and sulfur and magnesium shows up as well. All of these minerals that we need a small amount of in our diet are incorporated into the molecules here. Again, remember, this is in the inner mitochondrial membrane. So here it is showing you how the free energy in the electrons decreases as we pass the electron down the chain, and we ultimately make a water molecule. Okay, water's a byproduct. Just like we made carbon dioxide when we did glycolysis in the citric acid cycle, what we want is to capture the energy of those electrons as they fall down the chain back to their ground state. We want to capture that energy so we can actually use it for work. And the only molecule we use for powering work in the cell is ATP. So what has happened is, remember, we said the electron transport chain, which is not represented here, as those electrons fall from a high energy level to low, it is pumping hydrogen ions against its concentration gradient back into the intermembrane space. Now, these protons don't freely diffuse across the membrane. They are pumped through that specific channel. So, chemiosmosis is the process by which these hydrogen ions that are now outside want to fall back down their concentration gradient. Remember, osmosis, as we talked about with water, is passive diffusion. No energy input is required. We also could see diffusion in a passive transport system of sugars across the semipermeable membrane. Possibly we could see it with salt or other molecules. Here we are seeing it happen with hydrogen ions. So, in the intermembrane space, the electron transport chain powers pumping the hydrogen ions back into that space so then they can fall down their concentration gradient, and that does not require any energy. But as it falls back down, you can see that we create ATP. Okay? That's done by ATP synthase. So here shows you all of the mechanisms. As you can see, as each electron is taken from an electron carrier, we are pumping hydrogen ions out. And then those same hydrogen ions will fall back down their concentration gradient to make ATP. Okay? So the electron acceptor is oxygen. The final electron acceptor, after the electron is passed across the electron transport chain through complexes 1, 2, 3, and 4, is oxygen, and we form a water. Notice that we're actually taking the electrons off the electron carriers, and we don't directly make ATP. We're taking the electrons off of the electron carriers to pump hydrogen across the membrane. Chemiosmosis, which we see over here, is where we make the ATP, and it's a passive process that allows the hydrogen ions to fall back down their concentration gradient, and it uses that energy to put our ADP and inorganic phosphate back together to form ATP. So, we can see that if you do glycolysis and fermentation that we make 2 ATP. Again, that is made by substrate level phosphorylation. However, if we use glycolysis, the citric acid or Krebs cycle, and the respiratory chain or electron transport chain, we make a net of 36 ATPs. Notice that we only make 2 ATPs in glycolysis. we make 2 ATPs net in the citric acid cycle. But then we take all of these electron carriers that we made in the various steps, and all of those, each electron carrier will account for 3 ATP. So as you can see, we have 2 plus 2 plus 6, that's 10, so we make 12 total electron carriers. 10 of them are NADs, which we can make 3 ATPs each, and FADH2s, we can make 2 ATPs each, so that is a total of 36 net ATPs. Remember, we had to use 2, so we have to subtract those off. So, here again is a summary. We have glycolysis, where we make ATP by substrate level phosphorylation. We have the citric acid cycle, where we make ATPs by substrate level phosphorylation. But our main carrier of the energy as we break down glucose is the electron carriers that we see up here that are all going to the electron transport chain, and then through chemiosmosis, where the hydrogen ions fall back down their concentration gradient, we make lots and lots of ATP. Now, we capture in the form of ATP about 40% of the energy in a glucose molecule. That's fair. That's pretty efficient. It's definitely more efficient than fermentation. Now, you may be thinking that, why isn't it 100%? Well, think of it even this way. A car uses a energy-rich substance to power the motor. And that transfer of energy as we burn that fuel is not 100% efficient either. Some of the energy is lost as heat. And that is true with living systems as well. Some of the energy in that glucose molecule, as we break those bonds using enzymes, is lost in the form of heat. The rest, about 40% of the energy, is captured in the form of ATP. Okay, we've talked about the overall equation of cellular respiration, the 3 steps. It's very important that you understand some differences here, okay? First, fermentation and cellular respiration are both oxidizing electron carriers. And they are also capturing some energy in the form of ATP. When we have aerobic cellular respiration, the final electron acceptor is oxygen. When you have fermentation, we also have a final electron acceptor, and that final electron acceptor is an organic molecule. So it's going to be an acid or something like that that contains carbon. Your book talks about it a bit, but we can also have anaerobic cellular respiration, meaning that you can have an electron transport chain that is more efficient than fermentation, but it happens in the absence of oxygen. So clearly oxygen cannot be the final electron acceptor. Sometimes it's sulfur, sometimes it's a nitrate. It is another substance. It is not an organic molecule, but it's also not oxygen. It is also close in efficiency to aerobic cellular respiration in that it uses an electron transport chain and chemiosmosis to make ATP. So, all the food that you eat, you may think, what in the world do we do with proteins and fats? It is true that sometimes we break apart proteins into amino acids and use those as building blocks for other things, as well as for fats. But proteins, carbohydrates, and fats can all be used to make ATP. you can see that proteins can be broken down into amino acids, and those amino acids can have their amino group taken off, and they can then be converted into pyruvate acetyl-CoA or another intermediate in the citric acid cycle. Fats can be broken down into glycerol and fatty acid, and those can easily have their carbon, hydrogens, and oxygens rearranged and be made into glyceraldehyde or acetyl-CoA. In both cases, they can contribute and allow these three processes to harvest the energy from the bonds in those substances. So, I'm putting this on a discussion board. Please make any comments or questions below about things you still don't understand about the catabolic processes of glycolysis, Krebs cycle, and cellular respiration as you find them in Chapter 9. You can also ask these questions in class when we return from spring break, and I will talk with you soon.